We have seen that microtubule and actin monomers, they are very, very well conserved. That's not the case with the intermediate filaments. I can show you actually, physically show you different intermediate filaments that have formed these specific structures. My nails, the hair, they're all made up of specific type of intermediate filament. So there's a significant amount of diversity in intermediate filaments. Let's talk about these different types of intermediate filaments. The central helical domain of these different types of isoforms of intermediate filaments is similar. The differences are in N and C terminal globular domains, which vary significantly. We have seen the structure of intermediate filaments in the previous module. Different families of intermediate filaments are expressed in different cell types. The most diverse intermediate filament family is that of keratins. There are about 20 different keratins found in different types of human epithelial cells, and about 10 more that are specific to hair and nails. A single epithelial cell can produce multiple types of keratins, and these different types of keratins can co-polymerize into a single network. Here we have two micrographs. Let me talk about the micrograph which shows these long strands, the green strands. These strands are basically keratin filaments, which is a type of intermediate filament. And these filaments, if you look at them, they seem to be transversing they seem to be going from one cell into another cell. The way we can figure out where the cell boundary is by this lightly stained blue line, which is basically the boundary of the cell, one cell, the plasma membrane. It seems like that these filaments are going from one cell to another cell. That is actually not the case, since we have already studied cell adhesion. These filaments, basically filaments from one cell, intermediate filaments from one cell, terminate at special junctions called desmosomes, which are clear here in the electron micrograph. So these are the intermediate filaments of one cell, and the adjacent cell also forms a complementary desmosome, and the filaments of this cell terminate in this desmosome. So if we look at it from a distance, it would look like that these filaments are going from one cell to another cell. However, in actual fact, that these filaments are linked together through these special molecules, which we had said are cadherins. So they are linked together from one cell to another via these sticky molecules called cadherins. So this is basically, I just wanted you to appreciate the extensive network of intermediate filaments special type of intermediate filaments called keratin filaments in the epithelial cells. Now, let me tell you a few more things about about intermediate filament diversity. Every keratin filament is actually made up of equal mixture of type 1, which is acidic, and type 2, which is neutral basic keratin chains. These form heterodimers, two of which can then join to form the fundamental tetramer, which is the subunit for intermediate filaments. Cross-linked keratin networks held together by disulfide bonds can survive even death of the cells that formed it. For example, the coverings of animals, the outer layer of skin has hair and nails and claws. They can outlive even the organism that produced them. The diversity in keratin is actually also clinically useful in diagnosis of epithelial cancers or carcinomas, as particular set of keratins is expressed gives an indication of the epithelial tissue in which cancer originated. And it is very useful in diagnosing and treating different types of cancers. Now I'm going to show you a chart which talks about different, which shows you different intermediate filaments. In nucleus, we have lamins A, B, and C. They form the inner lining of nuclear envelope. Vimentin-like intermediate filaments. Many of mesenchymal cells from mesenchymal origin have these type of intermediate filaments. Desmin in muscle cells, that's the type of intermediate filament. Glial fibrillary and acidic protein has also vimentin-like intermediate filaments. They are found in glial cells, astrocytes, and some Schwann cells. Preferin is found in some neurons, along with other type of intermediate filaments. Epithelial cells have keratin type 1 acidic or type 2, which is basic. Type 2 is, and its derivatives are found in hair and nails. Neurons have NFL, M, and H type of intermediate filaments. Intermediate filaments are very stable, and as I've mentioned, that these structures made from these intermediate filaments, they can survive even after the organism that produced them has demised.